Good morning. Welcome to NISS Merck Meetup on Statistical and Deep Learning Methods for Biomedical Imaging. So my name is Pei Tao. I'm a biostatistician from Merck. And today I will be your moderator today. So first, I want to thanks to the NISS for hosting this wonderful event. We will have the opportunity to explore this vital topic. And so you will be hearing 3 great presentations from experts on this timely subject. So our speakers are John from Merck. Maybe we switch it out. So Andrew from the Castlewood Reserve University and Dr. Hongtu Zhu from the University of North Carolina. And so each speakers will have 15 to 20 minutes. So we will defer all the questions to the end section. And so we will have the 30 minutes, Q&A section at the end. So please use the Q&A feature at the bottom of the screen to ask questions. So I will read the question, to the end to the presenters so they can clarify, comment, answer the questions. So before the great presentations, there's some housekeeping, notes. So Jim and Megan are the hosts for this session. They are from NISS. And so I think many of you already know the NISS is a nonprofit organization which promotes research. So they hosted many, seminars, workshops, and so if you are interested in the upcoming events, please visit the NISS.org. So there's so many interesting topics and wonderful events there. And so today we have about 200, attendees. So I think with that, I want to stop talking. So let's please welcome our first speaker. I think Andrew Chen will be our first speaker. So when I introduce you, can you please, put on your slides? Yeah, great, thank you. So, Dr. Andrew Chen is an assistant research professor at the Center of Computational Imaging and Personalized Diagnostics at Case Western Reserve University. And he is a senior research scientist in the Precision Oncology Center at the Lund University Hospital, Switzerland. So for over 10 years, he has applied computer vision algorithm to digital pathology images. One of his areas of expertise is in leveraging deep learning to build computational models to aid pathologists in many common tasks. So Dr. Jen has a 2016 Journal of Pathology Informatics paper entitled Deep Learning for Digital Pathology Image Analysis, a comprehensive tutorial with selected use cases, received over 30,000 views and lay out a generalized approach via open source code and data sets to facilitate the development of next generation of data scientists. So his tools, HistoQC, a quality control pipeline for digital pathology slides, received the Innovation Award at the European Congress Digital Pathology. So with that, so it's all yours, Andrew.
Thank you. So can you hear me and see my screen okay?
Yes.
Okay, fantastic. So my talk is on computational pathology and how we're using it to address precision medicine. I have 3 topics that I'd briefly like to touch on. So I'll provide a motivation for computational digital pathology. We'll look at some research applications of that technology, and then some of the tools that we're releasing into the open source realm. So this talk focuses on what we call computer-aided diagnostics, or CAD for short, which is essentially using algorithms to help clinicians analyze data. This can, of course, be any type of data, such as fMRI, MRI, histology, X-ray, CT. My specialization is in histology, and this is an example on the left-hand side of a histology slide that we typically analyze. Why do we claim that CAD is useful? Essentially, it has the ability to improve the efficiency and robustness of medical diagnoses by making them essentially faster and more reproducible. We also want to be able to leverage the vast amounts of data that are already in existence, in particular noting that every day there's more data being created at an ever increasing rate. So there's more hospitals that are transitioning to digital workflows. As they're doing that, they're purchasing more scanners. So you kind of see this dual improvement or this exponential growth in the amount of data that's being generated every year in this field. And we'd ultimately like to be able to use that. So how can we actually use this data? Essentially, we want to perform data mining to identify trends. These trends are going to be subtle image patterns that may not be visually discernible or may be fairly difficult for a human to quantify. And I have some examples of that today in this talk. We'd like to build systems to aid but not replace doctors through decision support. This leads to the long-term goal of precision medicine. Precision medicine here essentially being providing the optimal treatment to a patient based off of their specific disease presentation, based off of large amounts of retrospective evidence. So we can essentially try and identify patients that are highly similar that we've treated in the past. We know that their treatment was successful, and we'd like to reapply that treatment to patients going into the future. There's a difference between research and clinical CAD applications. A lot of the clinical applications are things we're seeing now enter into the mainstream. These essentially involve recapitulation and automation of existing processes, such as detecting cancer, trying to do grading, counting, area estimation tasks. These are essentially tasks that are fairly laborious for humans to do. There's also issues with reproducibility. And if we can go and provide better quality in that regards, we're able to improve patient care through better definition refinement and better specificity. So instead of saying there's about 80 mitoses or there's approximately 50% area of something, we can say there's exactly 147 mitoses. These are the locations. The area is exactly 38%, and this is exactly where that area is. So just by improving that specificity, we've already shown that we can better stratify patients. Then there's something that we would consider research applications where we extend beyond what is currently done in clinical practice to try and identify novel biomarkers or novel features, and hopefully to identify increasingly specific subtypes so we can improve the kind of precision medicine that I mentioned on the previous slide. We'd like to connect this with some type of biological elucidation. And here we'd expect to see patient improvement in terms of augmentation and new insights. So it's not just using the same grading schemes that currently exist, but extending those grading schemes into the next level or the next generation of features. And we have some examples of those as well today. Most of my research focuses, we'll say, on the second part here. And we see that there's probably about a 10-year difference between the research applications and actually pushing them into clinical practice. So what does this experiment typically look like? We have large whole slide images. We extract regions of interest of them. From there, we'll do something like nuclear segmentation. Then we'll extract features from there, such as size, shape, eccentricity, et cetera, and we'll put it into some type of machine learning classifier. We'll do this for every other feature and every other histologic object of interest that we know about, some that are part of grading schemes, some that we're also interested in investigating. But ultimately, the end result out of that machine learning classifier is we'd like to predict either that outcome, a subtype, or another clinical variable of interest. We've heard a lot about deep learning, which has resulted in a large paradigm shift. This paradigm shift was essentially fueled by computation. We have access to large numbers of cloud computing resources. GPUs are fairly inexpensive now, so we can really afford to chug through the data that we have. Storage has become significantly cheaper, where we can actually afford to store thousands of whole slide images, where previously that wasn't possible. And as I mentioned, data. So we have large public repositories that are being created, such as the Cancer Genome Atlas, as well as I mentioned, hospitals are increasingly buying scanners, so they're naturally generating more and more data for us to analyze. As a result of what we'll call the deep learning revolution, we've replaced every single one of these components here in this blue box with a deep learning algorithm instead of our previous handcrafted approaches, while we've left the right-hand side intact, so we retain highly interpretable, visual verifiable features for our classifiers and biomarkers. So why do we use deep learning? Ultimately, deep learning is faster than building handcrafted features. So for example, building a nuclear segmentation algorithm was previously considered to be on the order of a PhD thesis on its own and would take about 3 years. Now, using deep learning in a similar annotated data set, we can now generate a superior classifier in about 3 hours. So we really see these 2 orders of magnitude speed improvement that were not previously available to us. And I think a common theme you'll see in this talk is this idea of magnitudes of order improvement in terms of speed or specificity. Deep learning also shows great robustness in terms of presentation and noise. An algorithm is happy to look at 1,000 slides and try and optimize across that. It's really not possible for a human to look at 1,000 things, keep them all in memory, and try and figure out the optimal, we'll say, model or parameters between all of those. At the end of the day, we came to realize that we don't really care how we get the pieces. We don't care how you segment the cells. We only care that once we have all of these objects, can we extract meaningful features from them and what we can actually do with them. So as mentioned during my very nice introduction, is that we released in 2016 a paper with the data, tools, code, tutorial to basically recapitulate every single one of the use cases that I mentioned on the previous slide. And that's available on my blog at andrewjanowicz.com, where we also keep it up to date with new and improved workflows. So you're happy to take a look and download them there. One use case I'd like to touch on is this concept of epithelium segmentation, where we see the original image, we see the overlay that a human pathologist has provided for us, and the right-hand side is the output from a deep learning classifier. Here we were only interested in the cells, but it's easier for humans to also include the white area. Now, while the bottom one looks very concordant or accurate, whatever metric you'd like to use, if we look at this top example here, we see that there's fairly low concordance. And in fact, it's about 60% accuracy or 60% concordance. When we brought this back to the pathologist, he started laughing and said that the result from our algorithm is more precise than what he gave us as input. So we can actually see that there's little pockets of stroma that are not highlighted here in green that our algorithm has successfully identified. He pointed out that it would be impossible in his work week to actually provide this level of specificity because it would take him hours per image, and he just doesn't have the time to do that, while our algorithm is able to do that within a few milliseconds. So again, we see these 2 orders of speed improvement while also having some increased specificity. We show that this approach is generalizable to other histologic primitives. In Kidney International in 2020, we again released 125 different biopsies that contained over 30,000 annotations across 6 different histologic primitives, and showed that this approach basically works across many different types of stains. All that information is also publicly available. Now we can look at some research applications. I mentioned in the beginning, we have this clinical desire of trying to recapitulate what is already being done. We know that in prostate cancer and predicting biochemical recurrence, there's a pattern called cribriform, but unfortunately, there's very low concordance between experts of what is or is not a cribriform pattern. What's interesting here is that we can use a computer to just essentially remove all of that subjectivity. So we train an algorithm to identify the cribriform pattern and subsequently go and apply it to a large cohort. And we show that in particular, in the Gleason grade 2 patients, which are either treated or given active surveillance, we can actually identify those which are at higher risk for recurrence and warrant additional screening. Now, the interesting component here is that although we're biased towards this single pathologist, only a single pathologist has provided annotations, the value here is that we can actually go and take this algorithm and copy and paste it across sites. So we can deploy it on multiple sites essentially for free, while we can't copy and paste a pathologist. So we now have something that's highly reproducible and something that is algorithmically also inexpensive to produce because the computation has become cheaper as well. We show that this concept also applies in predicting heart rejection. We see that there's a high inter-observer pathology agreement, and the algorithm that we built is statistically non-inferior. Again, this is interesting because although we can't say it's better than a group of pathologists, we at least know that it's deterministic. So we can consistently go deploy this on multiple sites and obtain the same values in a robust manner. This is kind of the next generation looking at feature extractions. So here I like this example because it's something that's difficult for a human to quantify. So here we identified cancer cells, that if cancer cells are highly entropic in their direction, that it's associated with more aggressive forms of prostate cancer, while if the cells are facing in the same direction, it's associated with a less aggressive form of prostate cancer. This is interesting because, first of all, it's not an easy pattern to identify, but now that the computer has identified it for us, we can very easily go and visually verify it. Secondly, we can appreciate that a human is not really designed to look at 1 million cells, estimate their entropy, and then come up with a single reproducible value between patients. Well, an algorithm is very happy to do this. So there's a value to using a computer algorithm, and this is one of the times we should leverage those things. We've shown that if we add that feature on top of things like nuclear shape, morphology, and eccentricity, we can start to stratify estrogen receptor positive breast cancers in terms of their survival beyond things like T-stage, chemotherapy grade, histology grade, and our features are actually more stratifying for those patients. We can additionally go and predict therapy response, right? So we don't want to identify just the patients that have an aggressive disease. We want to try to find a therapy that works well for them. This work here used multiple deep learning classifiers, first to identify where the tumor is, to identify the tumor cells, to identify the lymphocytes. From that, we identified tumor infiltrating lymphocytes, and then we extracted 5 very interpretable features. And by connecting these features, we showed that we're able to better identify the patients that are going to respond to immune therapy over just TIL density alone, which was previously considered to be a potential biomarker. To facilitate this research, we have a U01 from the NIH to develop tools and release them into the public space. A major concern that we found is ultimately machine learning, and deep learning in particular is subject to quality control problems. So this is a very clear garbage in, garbage out type situation. This previously wasn't insurmountable because we had maybe 10 slides. We would just look at them and remove the bad quality slides. When we're generating thousands of slides a day, this simply is no longer feasible. And as well, we've shown in a recent work that it's actually not reproducible. So we showed 330 slides to 3 readers and asked them, is this slide good enough to analyze? And you see that there's actually very low concordance. This is fairly alarming because if people start with different input to their experiment, even before the experiment begins, of course, they're going to get different output. We show that using our tool, we can raise the concordance of that in some cases by an absolute additional 25%. So what is this tool? The tool is called HistoQC. It essentially identifies artifact and quality control concerns. It also helps in identifying batch effects. This tool is free and publicly available at histoqc.com. If you're someone that's involved in the imaging space, we have an extension into the imaging space as well, available at this GitHub link, which is available on my final slide. We also have a tool called Quick Annotator. The idea here is that it becomes very challenging to annotate millions of cells. So we built a tool where the user starts to annotate cells here. You can see on the right-hand side. At the same exact time, a deep learning classifier is trained and starts to make suggestions. The user can either accept those suggestions or refine those suggestions. And as they're refining those suggestions, the deep learning classifier improves and this iterates. So we can now go and start to annotate large quantities of data, highly precise, similar to that epithelium stroma use case I showed before, without the time burden for a human. We've showed that improvements here, again, 2 orders of magnitude for some of these use cases. And because it's deep learning based, we can in fact use it across multiple different types of histologic primitives, from cells, tubules, epithelium stroma segmentation, et cetera. That tool is also free and publicly available at quickannotator.com. And the last tool I'd like to touch on is a tool called Patch Sorter. So once we identified a bunch of cells, we often want to apply labels to these cells, or it doesn't have to be cells. It could be any objects, for example, glomeruli or tubules, or really anything that we want to identify subcategories to. So what Patch Sorter does is it takes similar looking objects and it beds them near each other using a deep learning algorithm. So similar looking things are plotted near each other. Here, the user is able to go and lasso these objects. These objects then appear on the right-hand side. It now becomes much easier to visually review because all of the similar looking things look similar, and our brains are designed to spot differences. So we can quickly review here and say, yes, all of these are not lymphocytes, and apply at a group level the label once instead of having to interact with each cell individually. At the same time, a deep learning classifier is being trained to purify that space and better try and stratify and pull it apart. So here we can see now, if you remember, the blues and the reds were kind of mishmashed before. This is no longer the case. We highlight the blue region. We see all of these very clearly look like lymphocytes, hopefully to a pathologist. And they say, wow, this is amazing. I just need to click the button once, and they've now assigned a bulk of those labels in a single go. We've shown that taking this approach, we experienced a 371% efficiency improvement, and this is on an early beta. We're hoping to get it up as well to the 2 orders of magnitude that we see in Quick Annotator in later versions. So I think that brings my 15 minutes to a talk, to a conclusion. Here's my email address. If you're interested in any of these tools or collaborating, please, of course, feel free to reach out to me. My blog as well has all of the data and a lot of the code that is linked into my GitHub. And the tools that I mentioned are free and publicly available at these URLs. Thank you.
Thank you, Andrew. It's a wonderful presentation with so much information. Yeah, it takes time to, consume. It's very informative. I really like it. So thank you so much. We will defer the question to the end. Yeah, I see some questions coming to the Q&A, Q&A feature. And so with that, we want to move on to the second speaker, Dr. Hongtu Zhu. Oh, thank you for putting on the presentation. So Dr. Hongtu Zhu is a tenure professor of biostatistics at University of North Carolina at Chapel Hill and was DD Fellow and Chief Scientist of Statistics at DD Chuxing. He was endowed Baoshanjing Professorship in Diagnostic Imaging at MD Anderson Cancer Center. He chaired the Department of Statistical Cognitive Team and feature engineering with AI scientists and engineers on the development of innovative solutions for the world's largest large ride-hailing platform at DD Chuxing. He is an internationally recognized expert in statistical learning, medical imaging, analysis, precision medicine, biostatistics, artificial intelligence, and big data analytics. So he is well known. He is being elected fellow of American Statistical Association and the Institute of Mathematical Statistics in 2011. So he has published 300 papers, journal papers, including Nature, Nature's Genetics. So with that, I want to give the ball pass to Dr. Hongtu Zhu. It's all yours.
Okay, thank you. Thank you for the invitation. The title of my, because I only have 15 minutes, I go pretty quick. And the title of my talk is about biobank scaled multi-organ imaging genetics and beyond. And this is based on joint work with all the members of my lab and also my former students, Bingxin Zhao and Tenfei Ni. And also my colleagues, Yun Li and Stephen Smith and Jason Stein. And this is the contents of my talk. And I go to the first part. And actually in my lab, usually we do end-to-end research. And actually we do five different aspects of research. First one, we try to build the data set. The second one, we do deconvolution. Third one, we do the knowing. And also we do integration and do predictions. And just to give you the layout, how do we build the data set? And actually there's, in the last 20 years, there's a lot of public available data set. I think that's very important for medical research. And actually I systematically going through all these, most of these data set and find something I'm interested. And in particularly, I'm a neuroimaging and also genetic person. I'm mainly develop and systematically connect public available individual level data for over 50,000 individuals in order to build the largest database in this field. And in general, I'm interested in two important aspects. And one is basically anything related to aging brain and another thing related to brain development. And this is the data set that I currently have. And besides that, one thing we are working on is try to integrate the neuroimaging is try to help us to understand the complex interplay between the brain and other human organs and the underlying genetic overlap. That's one of the ongoing big projects we are working on. We try to integrate the brain, kidney, retina, and the heart together, and the basic systematic looking at the underlying kinds of causal pathway associated with this kind of different organs. And besides looking at the data, first thing we need to get to the data and get the data annotated in a certain degree. And the next question we try to do is try to process the data. We need to do deconvolution. And this is just to give you examples. Several papers were published in the last several years. And this is the one we finished this project for this year, and actually it has lasted for a long time. We developed a called a CLIP, is a clone and subclone structures and identification through the pairwise differences of polarization to distinguish the subclone structures. We tested the CLIP on almost 1,000 simulated samples generated by Broad Institute and all samples generated by using copy number profiles from actually patient samples. And this is the one paper we published in Nature 2 years ago. And actually we applied our method to the International Cancer Genome Consortiums. And we have these kinds of whole genome sequence data for over 27 samples. And we identified the clone and the subclone structures. And another topic we're working on is learning. Basically, when you have this larger scale data, and besides you need delousing data, reconstruct the data, and also you need to identify the patterns in the data. That's critically important. And this is related to diffusion weighted images. And you have the raw data, the diffusion weighted images, then you go to the processing pipeline, you get the imaging data. And actually you extracted like wide matter bundles, and you have the process of the data. The data is very complex in the sense of each subject after the wide matter reconstructions, you have over the more than several millions of the fiber bundles in the brain, in the 3D space. You need to do a lot of imaging reconstruction. Besides that, we need to map these the structural kinds of connectivity with the functional connectivities and put them together to build these kinds of the brain, the functional based the structural connectome atlas. Because that's the basically, if you do any of these kinds of the brain related research is very important to build the atlas so that you have better interpretation and also better scientific findings. And another thing is we try to when we try to build delousing, deconvolution, and also pattern learning. The next thing is we are trying to do the predictions. And this is one of these challenges we did almost 5 years ago. And actually, this is the H and E data. That's the first big talk about it. And we have these normal and the tumor subjects. And we basically we go from the raw data, how to do the pre-processing and also the feature extraction and to build these prediction models. And today's the main message here is to try to do integration with how to integrate the data across the different domains and how to really integrate together to find their relationship. And this is something I did almost started to work on this almost 10 years ago, and we're talking about the imaging genetics. And we try to, at the beginning, we are very simple. We're using the imaging to get to the imaging phenotypes and also have genetic kinds of the information. We try to correlate them together in certain degree. And then we're talking about the second part is big data in imaging genetics. And why do we do it? And actually is we try to use the brain imagings to capture the brain structure and the function changes associated with major brain related disorders and the normal development, and like Alzheimer's disease associated with the brain shrinkage. And why is the genetics are important and for the brain disorders? Because most of the major brain disorders like AD are heritable complex trait or disease. Actually, the genetic account for about 50% to 70% of AD risks, 75 to 90% of ADHD, 60% of 85% of the schizophrenia risk, and 80% of the ASD risk. And compare with the many other diseases, and actually this is the most of these brain-related complex traits, and many genes are contributed to the risk of the Alzheimer's disease. They have these kinds of specific pattern called polygenic genetic architecture. That means the genes are basically the effects are very small but non-zero contributions, and we call these non-sparse weak kinds of signal patterns. And one thing is when I And then many people ask me, why do I do the imaging genetics? The key things is to think about what's the relationship between these genes and the disease. And also you think about, and also confounded with all these environmental and social and psychological factors. And actually we try to build these kinds of dynamic kinds of path diagram. Is basically it goes from the genes, the molecule, and the brain cells, and the structure and the functions, and to the disease. Okay, these are systematically, and also until you can diagnosis by using symptoms like a behavioral test. And all these things, the environment contribute a lot through these kinds of process. That's it. We try to understand, and we try to understand the whole picture about these path diagrams. And the imaging analysis is only a small part of this kind of whole path diagram. That's the reason we try to do this, link all the things together. That's very important. You're looking at many complex disease, even cancers, you can do the same, something similar. And actually one thing is, and compare with the first talk, they mainly talking about H&E type of the images. Most of the images I'm working on is many either from CT and also from MI. And actually and because I have been working this area almost for 18 years, and I'm working close with many people in the areas, so I try to harmonize the tools and the pipelines to consistently generate the full spectrum of the neural imaging features from the brain. And that's basically, this is generate a systematic, a series of kinds of pipelines about our group. And just to give you simple ideas, it's basically like the brain volumes and the shape, and you can basically calculate the subcortical regions and the subregions and the size and the shape analysis on that. Also, you can do the cortical surface, and you can looking at the cell distribution and other information. And this is the white matter kinds of microstructures relate to the white matter bundles. And you can extract the diffusivity measures along these bundles. And actually, although these kinds of diffusivities are sensitive to specific type of microstructure changes and have been widely used for clinics. And also this is related to function of MI, because this is the related resting or task function of MI. And this is the related to, we use this ICA based method to form the 76 function region that generates these kinds of 1701 functional connectivity traits in order to characterize major functional brain regions and their connectivities. And actually, if you're interested in this area, my friends and Paul Nishan and Paul Thompson, they wrote a paper, review paper about this topic called the brain imaging genetics is a learning problems. You want to get into some of these. And the problems usually we are interested, these are 3, 4, 5 different aspects. One is basically we develop theories and all the method tools for the brain imaging genetics. The first things we're looking at the multiple biobank integrations, and you have to address the heterogeneities in global population. Also you needed to do omics integrations, and also we needed to develop new computational tools and for these dense and the weak signals. And also we have to deal with the method for dense signals. And we have a series of papers along these lines in the last 10 years, and you can get a sense of what we are doing. And the last part is about a series of clinical paper published by my group. And actually, in the last several years, I'm mainly working in a high-tech company. And then actually most of my students and did all this work. We developed this called the Brain Imaging Genetic Knowledge Portal, and they're called the BKP.org. If you're interested, you can visit our website. We aim to build the base knowledge databases of neuroimaging genetics, and you can find a lot of information inside. And actually, the current is a searchable database for over 1,593 plus neuroimaging traits across the different imaging modalities. And this is the one that typical examples related to the network load 25 and the load 20, and this is amplitude traits and for the function of MI. And also we have this GWAS summary statistics. The full set of GWAS summary statistics have been made free, available to the research communities. And actually, there's a lot of download for our summary statistics if you're interested. And this is the design for our recent paper published in Science. This overview of Enigma. This left side is our GTI pipeline. The right side is our design, the GWAS. And then how do we integrate the data from the different studies to do validation and also across the different phases. And this is one of the first major findings related to the genetic architecture of the white matters. We observe 109 novel genomic regions associated with white matter microstructures. And actually for white matters, the heritability is around 45%. And actually, this is basically for 25 known genomic risk regions of the GBM, is brain cancers, and 11 are associated with white matter microstructures. And this is also genetic collocation among the vascular risk factors like obesity, diabetes, high blood pressure, and also associated with the white matter microstructure and the stroke. And also we show this stronger genetic correlation between the white matter microstructure and smaller vessel, the stroke subtype. And also are able to do this kind of enrichment analysis in brain cells. We are able to identify brain cell type where the genetic variation due to changes in white matter connectivities. And actually we find that the 49, the complex trait that significantly enrich the genetic region influence the white matter microstructure such as stroke, schizophrenia, ADHD, bipolar, and Alzheimer's disease, T2D, and high blood pressure and coronary artery disorders. And also we do the functional FMI papers. This is one of the papers we're going to be publishing in the Nature Genetics pretty soon. This is basically, we were looking at the triple network models of psychopathologies, and we're looking at these kinds of senior network, central executive network, default model network. And these are highly related to the brain, major brain disorder like AD, Parkinson's disease, and major depression orders and others. And this is highly heritable. And then this is basically the genetics of the triple network compared with other function network like motor and visions. And this is the additional genetic signals and associated with the function FMI and data. And also we see the collocations between the brain functions in the default mode and the central executive network with the Alzheimer's disease and the schizophrenia. And the ApoE is one of the most of the families has a stronger genetic relationship with brain functions than brain structures. This is why the quite surprising findings. And also we see the collocation at these particular regions on chromosome 17. And actually these particular regions are associated with neurological disorders, psychiatric disorder, education, cognitive abilities, and psychological traits and alcohol use disorders. And this is the additional one papers we just submitted is related to kinds of refined kinds of postulation for the resting and the task function of my kinds of postulation. And we have this pipeline to how to process the data and how to do the imaging genetics for this refined kinds of postulation. And also we apply these kinds of things to these kinds of signal extraction and do imaging genetics on this data set, and we'll get a lot of signals inside, and these additional findings associated with that. Given the times, and actually you can visit our website, and this is the series of publications by my group on this topic. In the last 5 years, I mainly focus on processing the data and trying to write the clinical papers these days, basically, and that's really important for us. And also recently we shift a lot of effort on these multi-organ images, abdominal, brain, and heart, and other images like eyes, face, knee, and hip. And actually we try to process all the data from the different studies. And if you're interested, and welcome collaboration. This is one of the recent papers where we try to link the brain with the heart. And there's the multi-organ images from 40,000 subjects. We share this genetic influence between the heart and the brain structures. And this is the knowledge portals related to heart imaging genetics. And if you're interested, you can download the summary suggestions for that. And this is all these ongoing research in our lab, including kinds of causal relationship and also optimal models, and identify the best practical strategies and pipelines, and also try to model the brain changes, the genetic effects across lifespan and align and integrate the different imaging modalities. And finally, I want to thank all these funding agencies and also my students and my former students. And also in particular, I want to acknowledge the UK Biobank, because that's without their generosity and nothing would happen. Thank you.
Thank you, Dr. Hong Tu-Zhu, for this wonderful presentation. It's covered so many contents for this extremely important and interesting topic. It's important for not just for pharmaceutical company and for the public health. It's really nice. Okay, so now, John, are you there? Okay,
Yep,
and now we
can
can hear you. Yes, I just want to check. You have some technical issue at the beginning, so good. So now you are connected. So yeah, please put on your slides, and I want to introduce you. Dr. John Kang is a senior principal scientist at Merck. He has expertise in performing bioinformatics and statistical analysis with proteomics, microRNA, mRNA, SNP data for both exploratory and clinical development purpose. He had years of experience with all stages of clinical development process for biomarkers, including assay quality control, drafting SAP, supporting SOP of clinical development lab, and performing statistical analysis, interacting with the scientists, bioinformation, doctors, and late stages clinical statisticians. So his specialties include translational medicine, data mining, pharmacogenomics, biomarkers, next generation sequencing, clinical biomarker assay validation. So with that, so John, it's yours, all yours.
All right, thank you, Pinning. So first of all, I just want to do a quick technology check to see if everybody can hear me okay and see the screen okay.
Yes, we can see.
Okay, great. Hello, everybody. Thanks for coming, and thanks for the sponsors of this organizer of this meeting for the invitation. Today, I will be sharing some of the ongoing efforts of Merck in this relatively new field of digital pathology, as well as sharing some of our perspective in terms of how digital pathology can potentially be useful in the pharmaceutical industry in terms of target discovery and drug development. So the previous two speakers have given a wonderful presentation more along the line of the technical aspect of digital pathology and how digital pathology can be used in various...
John, sorry to interrupt. Can you make it full screen? Is that possible to make it full screen?
Yes, let me see if I can do that. Okay, can you still see my screen?
We kind of see the...
Can you switch display settings at the top?
Display setting to switch.
Okay,
I At the top of the screen, there's a presentation symbol.
Yep. Let's see.
Or maybe you can let Jim to
Right. Yeah, Jane,
present.
maybe you can navigate the slide if that works for you as well. Yep. Thanks, James. So great. Sorry for the technology issues that I'm having this morning, but so going back to where I was saying earlier, so the previous two speakers have given a wonderful presentation in terms of how digital pathology can be used in various academic research applications. And my focus for the talk today will be sharing perspective of how digital pathology can be potentially used in pharmaceutical industry, focusing on more high-level survey of the methodology, as well as providing perspective of how the potential of digital pathology in the drug discovery space. The outline for my presentation today is as follows. I will first give a overview of the motivation behind pursuing digital pathology in the translational oncology space in the setting of pharmaceutical research. And then I would give a very brief introduction of our Merck's analytics workflow implemented in-house to analyze this kind of data. I will then talk about a few case studies, some future directions, as well as a summary and conclusion for the talk. Next slide, please. So in the past decade or so, one of the important topics that has emerged in the scientific community was how can we potentially predict the patient's response to a treatment given their biomarker profiles. And specifically in the space of translational oncology, there's increasing evidence that the tumor microenvironment, or TME, has been implicated as being important in driving these differential response patterns of patients to these ICIs or immunocheckpoint inhibitors. And very loosely speaking, TME, or tumor microenvironment, is generally defined as the interaction or composition or organization of different important cell types in the tumor ecosystem of the patient. These important cell types including the immune cells, tumor cells, and stromal cells, etc. Next slide, please. In the noble pursuit of trying to identify biomarkers that can stratify patients' response mechanisms in the past decade or so, there has been tremendous amount of efforts, both in terms of developing the dry statistical methods as well as developing the wet lab techniques in trying to quantify or understanding the tumor microenvironment using those traditional assays. When I say traditional assays, I mean things like RNA-seq, those things like GWAS studies, as well as epigenomics profiling, which focus on quantifying the abundance of the biomarkers. For instance, people might ask, what's the correlation between the expression level of PD-L1 protein and the propensity of patients to respond to treatment? However, one of the very important elements that's missing from simply focusing on quantitation of abundance of biomarker expression when trying to characterize the tumor microenvironment is we're missing a very important element, which is the spatial organization of these important players, such as genes and cell types in the tumor ecosystem. So to answer that question, the emergence of what pathomics or imaging-based omics has become more and more popular because it provides this additional information of not only how much or how abundant these biomarkers are, but how geospatially distributed they are in the tumor ecosystem. Next slide, please. So given this additional information that pathomics might bring to the table in understanding the precision medicine, there has been a growing popularity of digital pathology in the scientific community. So this slide is in a courtesy of our commercial colleagues at Merck. Just in the past few years, we have quite a few number of AI companies that specialized in performing pathomics or image analysis entering the space of digital pathology. And just to name a few, with Alkins, PathAI, Snaps, etc., that claim that they can do a pretty good job in performing automatic digital imaging analyses. And in addition to those more specialized AI companies, there has been more and more presence of big pharmaceutical industry players as well in the space. And some of the early players in the digital pathology space, including the AstraZeneca, Novartis, and Bristol Myers Squibb. And more recently, Merck has also devoted more and more research trying to understand whether or not there's a true potential of digital pathology in the realm of drug discovery and development. Next slide, please. So given that there's such a hype or popularity of digital pathology, as biostatisticians, we're facing the increasing burden or pressure from the senior management trying to understand what's the true utility of this digital pathology, or what's the bottom line here in drug discovery and development within the pharmaceutical settings. So to answer the question, a cross-functional team within Merck was assembled, including my colleagues from companion diagnostics, bioinformatics group, as well as some of our team members in biostatistics, etc., trying to understand, and also commercial colleagues, trying to understand how useful the technology is for the development of the drug. So if the technology team deems that this technology is appropriate or has potential promises, and the pilot study will be planned and executed. So on the right-hand side of this slide, I'm kind of describing various phases for these pilot studies that the Merck has been using to evaluate the potential of digital pathology for our uses. And as biostatisticians, we play important roles in all phases of these pilot studies, including planning, execution, and validation. And because the AI, digital pathology, is a relatively new data modality, in all phases of the pilot study, there are challenges and statistical considerations that need to be taken into account when we execute each of the 3 steps of these pilot studies. For example, even the validation step, we have found through our experiences that a Bayesian go-no-go decision criteria is probably more appropriate to evaluate the true potential of digital pathology in achieving certain goals of our applications. But for today's presentation, I'll mainly focus on the second part of our roles as biostatisticians in evaluating this technology, which is the execution. So during the execution step of this pilot study, we often implement our internal pipeline for doing the analysis that similar to what the pipeline that the external vendor implements, and hoping to compare head-to-head and also to have a more objective assessment of the true potential of the technology. Next slide, please. So before I talk about implementing a deep learning pipeline for doing this kind of imaging analysis for pharmaceutical research, I mean, Andrew probably has already talked a lot about this in his talk, but I just want to give a very brief introduction of what kind of images, what kind of input data we're having for the data analysis. So the images commonly come in the format of a high dimensional matrix with rows and columns here representing the height and width of the image. In terms of the most basic units are pixels, and each individual number inside this matrix are the pixel intensities. So you can imagine that the input data, the dimension of input data will be the height multiplied by the width multiplied by the number of color channels on the image. And in our common practices, the most common data dimension we encountered in our daily data sets is in the range of billions of pixels. And this represents a very common setting of large P, small n setting. And the traditional statistical methods are not appropriate to handle this kind of data sets, because first of all, the data set come into 2 dimensions and there's inherent correlation both on the X and Y axis. And secondly, it's high dimensional, and most of the methodologies in the traditional statistical framework doesn't really apply here. So to answer these questions of how we can utilize these images and how to best leverage these images for our statistical analysis, usually the advanced machine learning techniques such as deep learning models that Andrew described in his talk will be used to analyze these data sets. So on the left-hand side, I'm providing a very simple toy example of how the convolutional neural network, which is one of the most fundamental type of deep learning model, can be used to extract different features of this number 7, which is the image, and then use the extracted features for the downstream association analysis with outcomes of interest. And on the right-hand side, you can see that as the network grows deeper and deeper, or more and more convolution operations are being performed, we can extract potentially more complicated features from the images, such as the face of the dog. And then these features can then be subsequently applied to our downstream association analysis again to evaluate the association between the image's features and the outcome of interest. Next slide, please. So one of the most common imaging types that we use, especially in the translational oncology space, is what we call the H&E images, which again has been mentioned multiple times in both Hoon Tu and Andrew's talk. And H&E images are acronym for the hematoxylin and eosin stain images, where hematoxylin is the stain for nucleus, which are the purple color on the image on the right-hand side. And the eosin is the stain for the extracellular matrix and cytoplasm, which are pink in color on the H&E stain images on the right-hand side. And the reason why H&E images are so attractive for the research purposes, and also as well as for the research development purposes in the pharmaceutical industry, is that first of all, they are routinely collected for diagnostic purposes. They're inexpensive, and the assay complexity is very low, which means that they are relatively cheaper to be collected. And in addition, they provide detailed representation of the geospatial arrangement of different cell types and potentially gene levels at the tumor microenvironments. The interplay between, for example, tumor cells, stroma cells, and immune cells can be captured in a snapshot fashion by H&E images. And finally, these images can be digitized into high resolution for more sophisticated and complicated quantitative analysis for the downstream association. And the pharmaceutical industry overall is interested in the H&E analysis for mainly three reasons. Again, these are my personal opinions, but the H&E can be quite valuable in terms of providing potential targets for target nomination in terms of drug discovery. And also they can serve as potential patient stratification factors in precision medicine, identifying which subgroup patients might more likely to receive certain treatment and benefit from that treatment versus other patients population might be more beneficial to receive another treatment, etc. And finally, because H&Es are so cheap and so low in terms of assay complexity, we're also evaluating extensively whether or not we can use H&E images as a surrogate of those established paleo-biomarkers, such as PD-L1 and tumor potential burden, which are typically profiled using those high assay complexity assays, such as next-generation sequencing, as well as IHC assays. So the H&E potentially can serve as a cheaper alternative to these more expensive and more difficult assays if indeed they can predict those biomarkers with high accuracy. Next slide, please. So in order to evaluate internally the potential of digital pathology in terms of the application we're interested in Merck, we developed this pipeline, what we call PrEP, to evaluate the pathomics data sets or the digital pathology data sets. The first step of a pipeline is to really perform the quality control using partially the package that Andrew mentioned, the HistoQC package, to identify the artifacts from the images and then remove those from the image prior to the downstream analysis. And then once the QC is performed, we will perform the RI detection to identify the tumor regions and predict on the tumor region the various cell types of interest. Again, the cell types here being predicted using deep learning models are the major players of the tumor microenvironment, and therefore is of the most interest, not only in terms of how much we have, they are present in this tumor microenvironment, but also the geospatial distribution of how they are distributed with each other in this tumor microenvironment, both of which could be potentially important in associating with the outcome of interest. So you'll notice that there are two separate branches coming out of the post-QC part of the pipeline. The first branch is what we call the HIF branch, which stands for the human interpretable feature. So that branch is particularly useful for us to understand what features are driving the outcome of interest by opening up the black box of deep learning models. And that branch is particularly useful for understanding the mechanism of treatment when we're talking about drug discovery. This bottom part of the pipeline is what we call the black box approach, in which case we care about the prediction accuracy the most, and not so much about necessarily mechanism of action. So in this case, the direct input for the pipeline is just the post-QC raw images, and then we try to directly correlate or associate that raw image with the outcome of interest using a deep learning model. So again, the main difference between the two approaches is one is with the mechanism of action in mind for drug discovery, and the second is with the prediction accuracy in mind that's potentially useful for generating a H&E version of a surrogate of the molecular biomarkers for potential digital companion diagnostic applications. Next slide, please. So for the remainder of this presentation, I will present two case studies. The first case study is using the HIF branch of the pipeline, focusing on understanding the mechanism or the opening out the black box approach that I described earlier. So to evaluate this part of the pipeline, we are trying to answer the question of how does the H&E images predict patients' treatment response to immunocheckpoint inhibitors? And we're working closely with our colleague in Discovery Oncology Department at Merck, and their main focus was trying to understand, for example, what's the usable features from H&E that can be helpful for them to answer potentially biological hypotheses they might have, and use that information to come up with new compounds or new mechanism that can explain the patient differential response patterns to our drug. So to answer that question, we use a large internal clinical trial study containing roughly 1,200 host-like images stained with H&E. And then we split the data set into training and testing. And then we use the HIF portion of the pipeline, the human interpolable feature of part of the pipeline, and then generate features using the predictive cell maps from the deep learning models. For example, things like what's the distance distribution between immune cells and tumor cells, and what's the co-localization patterns of tumor cell clusters, immune cell clusters, et cetera, et cetera. Those are what we call the HIF. And we try to utilize these human interpretable characterization of TME and correlate those with the treatment response of the patient, which is our outcome of interest here, and then trying to validate or correlate the model predicted clinical response propensity with the true response patterns in the independent validation data set to validate how well can the H&E model predict the patient treatment response to our treatment. Next slide, please. So here are some key findings from this pilot. On the left-hand side, is the line plot here is showing how does the patient response pattern or propensity to immunocheckpoint inhibitors decrease as the distance between the lymphocytes and tumor cells increase. And the two lines represents two separate clinical trials that we utilize or applied our models on. But in both trials, although the magnitude of effect are different, but the trend is similar in terms of we see that not only the abundance of lymphocytes are important in driving the patient's response to immunocheckpoint inhibitors, but also the distance distribution between tumor cells and immune cells that's important. And that makes intuitive sense because the closer the immune cells are to the tumor cells, the more readily attackable, if you will, the tumor cells are toward the immune cells, and therefore the patient might have a high propensity to respond to our immunocheckpoint inhibitor treatments. And the right-hand side is another features that was discovered to be highly associated with patient response mechanism, which is the co-localization of the tumor clusters and the immune clusters, which we call hotspot features, which is consistent with the literature in the spatial statistical literature that the hotspots between immune cells and cancer cells are positively associated with patients' propensity to respond to immune therapies. And the box plot on the right-hand side is showing that in the validation set, we're able to replicate that the proportion of the immune cancer hotspot is more upregulated in the responder compared to non-responders, consistent with our findings in the training data set. Next slide, please. So far I've talked about how we can use the digital pathology pipeline to predict a more, I guess, a promiscuous phenotype, in this case, the treatment response to treatment. In this case, the scientists more care about the mechanism of the action rather than prediction accuracy. However, we do have some other applications where the prediction accuracy is the main focus. And this, where my case study number 2 come to play. And the main question of interest that we want to answer is, can H&E images serve as a cheaper alternative to the more expensive kind of assay type generated biomarkers, such as tumor mutation burden? And just a very brief background here, TMB, or tumor mutation burden, is an FDA-approved killer biomarker for the ICI treatments. And however, the TMB profiling requires using the technology such as next generation sequencing, which is not only expensive, but also not readily available for all patients. And therefore, our question of interest now is is really, can we use the H&E images, which are cheap and readily available for all patients, to predict TMB, and with what accuracy? And if so, is there any potential to use H&E to replace the more expensive and complicated assay type of the NGS TMB profiling? To answer that question, we utilized a attention weight-based multiple instance learning framework, deep learning model, to predict directly from the images, to trying to correlate that with the TMB high and low status of the patients using the black box approach that I described earlier in the PrEP pipeline. And we're able to achieve a relatively high AUROCs, which is 0.82. And we're also able to open up the black box a little bit by looking at the distribution of attention weight on the original image and figure out that whether or not the most predictive regions are indeed the tumor regions. And upon confirming with the pathologist, it looks like the most predictive parts of the images are indeed the tumor regions, which is the most relevant part of the image that we've hypothesized to be TMB related. Next slide, please. So far I've talked about some of the powerful applications of how H&E can be used in some of the drug development applications. But we have to keep in mind there are major limitations of H&E images, mainly for the following reasons. The first major limitation of H&E images is that because of it's such a simple assay, it only stains nucleus and cytoplasm of different cells, the detection of certain cell types based on the H&E stains is not always perfect. So we ran into trouble of using H&E to detect myeloid cells. So again, it depends on the cell type. The detection of H&E might not be the most appropriate. And secondly, which is I think the most criticism came from based on our interaction with the scientist, is that the immune cell subtypes might carry the same morphological features under the H&E stain, but depends on which subclass you're looking at, they might carry completely opposite biological functions. So for instance, the macrophage cells, M1, M2, they look identical on the image under the scope, but one is beneficial to response and the other is detrimental to response. But you cannot really tell M1 versus M2 based on just H&E stains. The same for T regulatory cells and T killer cells. They look, again, the same under the microscope, but they carry complete opposite functions. And third limitation for the H&E is that it doesn't really have the single gene resolution we need for target nomination, right? Because, although, yeah, we can say the tumor cells and lymphocytes, the closer they are, the more likely the patient is going to respond. But what can we do with that information for our drug discovery? But that's why the single gene resolution might become important in the context of that thought. So my current effort, in addition to the digital pathology image analysis, is trying to integrate digital pathology image analysis with the more cutting edge spatial transcriptomics data, where we do have both spatial information and the single gene resolution on the same image. And the question now really becomes how to integrate the two data modality together for a more accurate depiction of the TME for the drug discovery purposes. Next slide, please. So to quickly summarize and conclude, the reason why pathomics and digital pathology has gained popularity over the past few years is because it does provide, to a certain extent, orthogonal information to the traditional omics layers, including RNA-seq, DNA data, epigenetics data, etc. And another element I want to mention here is that unlike, for example, in RNA-seq, etc., you don't necessarily have to use deep learning models, but for the pathomics data analysis, because of the unique data types and the unique challenges of this modality, we often have to use the non-traditional statistical analysis methodologies. And that's where the machine learning methods such as deep learning models become important in our day-to-day life. And I also hopefully convey this message that although it's challenging to analyze the H&E images, but there are potentials of the H&E images in the drug discovery space. And there have been both internal and external studies, internal and external with respect to Merck, that demonstrate the proof of concept that the pathomics and digital pathology could be useful and powerful in certain applications we're interested in. And finally, we have to bear in mind that H&E does have its limitations for the three reasons I mentioned on the previous slide. And ultimately, I envision that at the end of the day, we have to combine the imaging data with the genomics data, such as spatial transcriptomics, to have a more accurate depiction of the true TME architecture. And with that, I'd like to thank the audience for the patience, and thank you very much.
Thank you, John, for this excellent presentation. So you lay out, an introduction for the HIE deep learning pipeline, to classify cells and predict maybe the outcome. Okay, so now we want to move on to the Q&A section. So, okay, I see there's two questions. So first I want to maybe let the panelists, do you have questions to each other? Okay. So I see there's two open questions in the Q&A. I think they are for John. So the first question from Sean Rong, and how HIF are identified. For example, the feature of distance between immune cells and tumor cells based on the biological knowledge.
Yeah, that's a great question. Thanks for the question. So there are two main methods we compile our internal HIF. One is through the literature research.
Could I interrupt and ask you to turn on your videos?
Sure. Yep. Sorry. Okay. How about now? Can people see me?
That's good.
Yeah.
And the other speakers can also.
Okay. Fantastic. Right. So as I was saying, there are two main methods we acquired this potential HIF for our research purposes. One is through literature research. And you'll be surprised how many papers that talk about the potential HIF that people have been using in their applications, just in the past few years or so. And secondly, the second source of features we obtain are just through talking to our scientists. What are relevant features they feel like are interested or worthwhile exploring? But I mean, from a more computational angle in terms of how we calculate distances, it's really just, there's no magic around it. We just, you identify the nuclei location of different cells and then just calculate pairwise distance from there. But of course, you have to define things like what neighborhood to look at and how you define neighborhoods, but those are just the minor details.
Okay, thank you. And there's another question, I think it's also for John. How are human interpretable feature determined, predetermined by experts prior to running the algorithm? Did you start with distance immune cells to tumor cells as feature, or was it discovered by the learning algorithm?
Yeah, that's a great question. distance, so first of all, we do have a training set and test set, right? So we're not using the same data set for both training and validation. And in the training set, we perform various cross-validation to ensure that we're not overfitting the data. So having said that, so there are two levels of statistical testing. One is what we call the hypothesis testing, in which case we do have a very specified hypothesis from the immunologist. For example, does the distance between immune cells and tumor cells correlate with the response? With that single hypothesis, our primary hypothesis. But then I think the question really relate to multiple testing and things like that, de novo discovery. So we do assign p-values or register p-values based on our results from the training set on the validation data set. But I guess the key take home is, first of all, is that the discovery is done on the training set, and on the test set, there is a formulation of primary objective and secondary objective, where the primary objective is focusing on the more important question of interest, and the secondary objective has a de novo flavor to it, and multiplicity correction is performed.
Okay, but so the question, is it possible? So the same person, Kay, asked the question, but it's possible you are missing accuracy by only using predetermined HIF features.
Absolutely. So that's why we have two branches of the pipeline, right? Depends on the particular application of interest. So for the HIF application, the scientists really are interested in what are the interpretable aspect of the images that they can use for their research, not just prediction accuracy. So there has to be a trade-off between prediction accuracy and how interpretable the results are, right? And for this application, our scientists feels like the interpretability dominates the prediction accuracy aspect, and therefore we're using the HIF approach. But absolutely, you might be missing some important features. That's why if we have a large enough data set for training, we potentially can discover the black box-based approach and combine that with HIF to derive new potential HIF not covered by the literature. But given our relatively moderate sample size, that's the best that we can do at this point.
Okay, I see, Mia asked a question. Can you elaborate your question? You said the p-value of what test? Maybe you can type the question more complete. So actually, I have a question for maybe from the outsider of this research area. I was wondering, and I think that this question is maybe for Dr. Hongtu Zhu and other, presenters also. I think, since it looks like the deep learning, it's a very vital and, advanced machine and learning algorithm, and it helped, diabetic, it helps to identify maybe, for the early stage breast cancer, the lesion, or it's used for brain imaging, and it's used for diabetic, maybe retinopathy, right? When the doctor is not available where patient is. So I was wondering, there's so many talks and it's amazing, how this can be used for clinical practice clinicians. But I was wondering, based on your experience with how the clinical practice, the clinician, they adopt this. So do you think it's still a bench, or do you think it's to the bedside? So can you comment on that? How the public health and the clinician will adopt that, the new technologies, but maybe they don't understand what's in the black box.
Oh, I think deep learning is the tools developed to handle the pattern recognition problems. And there are three major pattern recognition problems in the field. and one is CV type of computer vision, and also natural language processing, all the type of things, and also NLP, okay, text mining, these kind of things. And because of all these data, they have the similar patterns. Basically, the orders of the data is not critical, because the data is basically the features, it could appear in a different location in the like a sentence or in a picture, okay? And this deep learning was mainly motivated by addressing the questions in this type of problems. And I think and actually they really push about the traditional statistical methods, like the built on the like a linear regression or harmonic analysis. And in all these kinds of traditional modeling techniques, they assume the data is aligned. Now, at least the feature aligned. Okay, basically you write a model like y equal to x based on linear model. Your x features has been aligned in certain degree. Okay. And but all the tradition, the deep learning type of the method is basically is the kinds of automatic kinds of learning, because the input data don't have to be aligned. That's the reason these days the traditional imaging processing techniques are not critical. And if you're really interested in doing prediction, they basically most time the people just dump the models into the deep learning pipelines, and they get then using the extractor features, because the deep learning automatically to align the data in the last layers and basically doing the prediction on the Y. And that's if you only for that purposes, I think these do have some advantages. But in order to train the models, and there are two ways to do it. One is you'll use a pre-trained models, and like you're doing BERT or other like a ResNet, this type of models. Then usually when you do the pre-trained models, that means they have a gigantic big data set. Then you use large resources that build kinds of models for the prediction to this or classification and this type of problems. And usually you have these prefixed models. That's the kinds of the features. For the new data set, you come inside, you can directly apply these pre-trained models to get the features in the kinds of common space. Then you try to predict. And I think that's the key advantages for these high-tech companies, because they have the tons of resources and also have to annotate the data, so that they develop all these tools for CV and also NLP. Okay, you'll see that all the major kinds of the recent big development in data science, many high-tech companies, because they have the tons of research. University doesn't have it. Okay, that's the key. And usually they just they have this connected data set, even come from different domains. The method that the data set, the model that learned from this big data set is still valuable for other data set from different kinds of domains. That's basically the pre-trained model. But if you really want to build the train this model by using your data set, if your data set is pretty small, it's not well annotated, usually the model you build is not very stable. Okay, practically may not be very useful for, in general, and the results can be easily modified and also updated in different studies. That means for a data set, besides the pre-trained models, you can build everything by your own, but you need a big data set and also annotate the data well in order to really train models. And also you need to have the computing powers to do all this work. Okay, I think for H&E and MI, all these kinds of different image modalities, I think at the down range, the main limitation is you have a good annotated data set compared with the CVs. But in many applications, what I do is basically apply existing the pre-trained model file based on like ImageNet to our data set and extract the features and do all the analysis, downstream analysis. And even for H&E, you can do the same thing. And that's very common. And I think if you want to skip the detailed training process and also build up, because there's a two difficulty. One is to basically build up the good data set. The second part is you need to train models. And I think but in the long run, you still need that for the one particular applications you really want to do that. Okay, but I think that all these models you're building is really push these kinds of the prediction models to the industries, because when you apply these things in the industry applications, and regardless you're in the medical science or other field, you have to basically produce the good, reliable results. Otherwise, nobody will use it, because that's a real product. I think that's basically the key things these days. Yeah. And that's important in practice. I think the deep learning really push the areas to the next levels, in particular for data science. You think about the last 10 years, what is the biggest achievement in data science? That's the deep learning, without doubt. Okay, that's the case. Yeah, they changed the whole the way we handled all the imaging, all the many data types.
And so Andrew and John, do you want to comment? Okay. There's another question from the Q&A question about the use of UK biobank data. Did you find that your data was in any way biased towards a healthy population, specific population demographics due to the nature of the people volunteering for it?
I think that's the case. I think we cannot avoid that. And there's the one thing you can specifically address the issues. One of my friend wrote a paper about the kinds of bias sampling issues for the UK biobank. And I think that's a good point that many people, some other people think about. Actually, there's a causal kinds of relationship. If you do this kinds of way, using volunteer data sets, collecting data from larger volunteers, set of volunteers, whether the funding is valid and in larger population or in different, the people with a different ethnic background. But I completely agree with that. But the key thing is this is the first and largest data set with almost complete information from all aspects. At NISS, this is the first time we have an opportunity to look at the data and find their relationship. I think you don't treat this as ending point. Is as other than this is a beginning point. I think that's the baby really give you the whole medical science basically the new way looking at the big data, the biobank data set. It never happened before. Okay, that's happened in the United States. Unfortunately, I have to say. At least one country, basically UK, they are willing to connect this large scale data that takes over more than 10 years and collect the data from all the aspects they couldn't imagine and really build this up. That's tons of work. Even all of us, the studies happen in the United States, I think it still takes another 10 years to reach that level they already did. Well, I think that's the whole. I personally very appreciate that they are willing to collect the data and sharing with the whole scientific communities. I think that they've truly revolutionized the medical science in a certain degree.
Yeah. Thank you. So I think the last question I saw from the, I think this is for John. Can you please explicitly mention the names of the test for which you are observing the p-values?
Sorry, to clarify, observing p-values meaning the kind of
okay, maybe Mia, so if you have the John's contact information, you can connect it to him to clarify your questions. So
please do.
okay, so it says all the question to Andrews, you answered it all through the QA. Okay, thank you. So I think to conclude today, thank you for all the speakers. That's wonderful, presentations, excellent work you did. And thank you for all the attendees. And this is, I hope you enjoy the amazing, presentation. And, for now, we want to sign off. And I hope everyone enjoy the rest of the day. Thank you.
Thank you.
Thank you.
Bye-bye.
Bye.